right, so last time we left you with salt bridges, and actually I have to say, given the part of the exam that I graded, it seems like you mostly got this stuff, so that's great, but I still want to go over it and finish it up. First of all, energetically important salt bridges are usually found at varied or partially varied positions. The ones on the surface that you see in crystal structures, most of them don't do anything. Again, they typically aren't all by themselves. They're part of a larger hydrogen-bonded network, which is in all proteins. All proteins have these, even smaller ones. How do you know it's a salt bridge? Well, the pKa's tell you. Again, most of you seem to have understood that. Basically, if there's a salt bridge, the pKa's will be in it. So energetically important one, the pKa's will be shifted towards ionization for both residues, although the amount that they're shifted isn't the same because of the different pHs at which you determine those pKa's. They're important. Knocking them out costs you a lot of kcals. So, and in fact, the paradox that I mentioned to you is that all of the folding energy of 2,4-lysozyme could be accounted for by the salt bridges. That should bother you because we know there's a lot of other things going on. But again, that's assuming those salt bridges are independent. And what's known is after you start knocking out one salt bridge, the other salt bridges actually get weaker. And that's due to the fact that the structure is now not nicely tied down, and those are not localized as well as before. And I'm going to leave you with a question, which is sort of a philosophical one. If you're interested, you can read about it. Are they always stabilizing? Are these salt bridges always stabilizing? There's a paper by Bob Sauer's group on ARC Repressor where they claim to find a salt bridge that is not stabilizing. But that paper, it's on my website, on the lectures. You can read it. It's got a lot of problems, but if you want to talk about it. So I would say the answer to that is yes. Okay, so let's actually look at some of these pictorially. So here's a T4-lysozyme salt bridge. These are all from T4-lysozyme, and it's the ARG95 ASP92 salt bridge. It's worth about 3 kcals per mole by itself. But these residues aren't just doing this. Okay, they're doing a bunch of other things. Let's look at this aspartate and where it's located. This is a helix, and it's in ribbon up until this point. This is the first turn of the helix, and look at those 3 unsatisfied amides. Okay, they're at the N-terminus of the helix, just like I said. And right above them, at the first position before the helix, is this aspartate. So this is also an end cap. Okay? Same thing over here. This is interesting. So we've got a helix again, but now we're not looking at the N-terminus, we're looking at the C-terminus. Why? Because these carbonyl groups are pointing at you. That's how you know it's the C-terminus. And they are also not satisfied, except when arginine 95 does it. So we have 2 hydrogen bonds being donated here, 2 hydrogen bonds being donated here, plus you have the salt bridge. Altogether, that's pretty important. And furthermore, this arginine is now stacked on this tryptophan. And all of that actually makes this residue particularly important. It's really at the center of a really large network. And if you take it out, the protein is destabilized by 5 kilocalories per mole, probably the net loss of almost all of these interactions. And interestingly enough, if you show guanidinium into the crystal, actually if you melt them in the presence of small amounts of guanidinium, less than a molar, the protein is actually more stable. So even the denaturant, in this case, is now acting as a ligand. If you do a crystal structure, the guanidinium group is just sitting exactly where the arginine side chain is. So that is really a guanidinium binding site, if you will. Okay? So there's others. There's another completely buried salt bridge in the middle of the protein, ASP10, ARG148. If you knock out ASP10 with just about anything but a serine, you get no protein. Okay? The protein doesn't fold at all. With serine, you can prep the protein, but you can't crystallize it. And that's because this guy, again, is right in the middle of a really giant network of hydrogen bonds. This is the surface of the protein over here. This is actually a residue in the catalytic site. And here's the chain of hydrogen bonds leading all the way to that residue in the center. So, pretty incredible. So I think the question you might ask yourself is, why are salt bridges there? Why are there polar networks? Why do you need to have them? Because you could conceivably replace all those residues with hydrophobic ones. The protein might still fold. And a few times that experiment has been done, that's what happens. The protein still folds. It's not quite as stable, but they're not completely unhappy either. And so this is something that we're going to address in the next lecture after this one. Okay, the last part of this is entropic stabilization. And really, the basic issue here is that when proteins fold, and again, I learned from reading your question 1 that you understood this, that the protein has many states available in the unfolded state and few states available in the folded state, and that is a huge loss of entropy. Okay, how big? Well, let's talk about it. So remember I told you a long time ago that you could actually calculate delta S, or at least the contribution of states to delta S, by taking the number of states in the final state over the number of states in the initial state, the natural log times R. That is the entropy difference between the 2 states. Now, the problem is counting these states is not so straightforward, but we can at least say conformationally how much entropy are we losing from the backbone alone when a protein folds up. And so you can do this calculation by saying you can estimate how many states you have. Remember I told you the number of states is sort of scales as a log of a power of the length. This is the rough version of that. And then we can also say that the folded protein itself has one state. And so if you kind of do the calculation for a 100 amino acid protein, you get about 54 kcals per mole at room temperature just for backbone entropy. And that's not even talking about the side chains, where we haven't really talked about it at all. But exactly what I've said going on in the backbone is also going on in the side chains. The reason we haven't talked about it is I don't think we need to beat a dead horse. It's the same deal. Yeah? Okay. So do be aware side chains are equally important. And things like arginine, that's an awful lot of degrees of freedom to lose for one side chain. So the side chain entropy is at least as big as the backbone conformational entropy. Okay. So if I wanted to then make this situation better and stabilize the protein, how do I do this? Okay. Now one way I could do it is I could say, well, this is one state. Why don't I just make it a lot of states? Okay. Whoa. That's interesting. Really messing up. Okay, I could just make it a lot of states. Suppose I increase the number of states available from one to more of them. So now that would drop the free energy of the folded state. This gap would get bigger. The protein would be more stable. Is that a good idea? Would it be a good idea to increase the number of folded states to stabilize the protein? Why not? Yeah. That's right. Well, increasing the number of states is, in a sense, you're right, making the protein misfold, because we've defined, the active form of the protein is really a singular or very small collection of folded states, and that tends to be the case. If we increase the number of those, most of them aren't going to be active. And so while you might make the protein more stable, you will probably make it less active. That's not very good. Okay? Not such a smart idea. Okay, well, we can't do anything to the folded state. What else can we do? What else can we think about doing? Well, if we can't work in the folded state, there's only one other state to work on, right? And that's the unfolded state. So maybe what we can do instead is we can say, don't let the unfolded state have so many states. That would be great. Free energy would go up, it would increase the gap between the unfolded and the folded state, and we stabilize the protein. The question is, how do you do that? Anyone have any ideas? How do you decrease the number of unfolded states there are? So the first thing, okay, yeah. I like your second idea better. Okay.
Yeah, you can make disulfide bonds so that you kind of limit the different directions the strand can go.
Yeah, so you can use disulfides. That's one way. In fact, that's what disulfides do. I told you this a long time ago, so I'm not surprised you don't remember. That disulfides, what they do is they limit the number of states in the unfolded state, because once you lock down two ends of a protein chain, the residues in the middle of that don't have as many degrees of freedom. What's another way to do that, based on your idea? Okay, don't make the folded state so flexible by mutating it. Putting in hydrophobic residues probably wouldn't help, because if they start to compact and do things, then that's right. Glycines have a big Ramachandran plot, right? Yeah. And what else could you do then? All right, proline. Yeah. So you can do all of those. Good. You guys are on it. So it's a great idea. Okay, nature did it first. It made disulfide crosslinks, and in fact, removing glycine and adding proline could be a good thing. So you can change the properties of the backbone by changing the amino acids, individual amino acid flexibilities, i.e., their Ramachandran plots, and then you can also crosslink the backbone. Let's talk about the first one. Remember, glycine has this nice big Ramachandran plot with lots of allowed regions. These guys, all the other amino acids except proline, have much smaller Ramachandran plots. So basically, going from here to here should really decrease the number of conformations available to the unfolded state, and that should give you an increase in free energy. But if you do the calculation, you have about 3.3-fold more states with glycine than you do alanine, and that roughly gives you about, at room temperature, about 0.7 of a kcal per mole stabilization, in theory. Okay? And it's even better when you go to alanine to proline. This one is a little bit harder for me to explain. I've never been able to find a Ramachandran plot for proline. So that's kind of an interesting question. But in any case, there are a lot more states restricted by having a proline there. And as a result, you can expect an even greater stabilization by creating proline. So going from glycine proline, in theory, could give you up to a 2 kcals per mole stabilization. So the real question is, how do you do this? How do you choose sites? Well, glycine's got a problem, okay, which is that you can do glycine-alanine substitution or glycine to proline substitution, but there's certain glycines that you can't do this with. And as we know, a large number of glycines, not the majority as it turns out, but a large number of glycines, at least some fraction, are in the left-handed phi-psi angles. If you put any other amino acid there, you're going to make the protein really unhappy. So that's a case of where you're going to mess up the folded state a lot more than you get for messing up the unfolded state. So that's the first thing. The second thing, if you're going to change anything to a proline, either a glycine or an alanine, you have to make sure that that residue doesn't have an amide bond that is doing something like in a helix. Okay? Now, we already know positions in a helix that actually are great for proline. Those are the ones in the first turn. Okay? Because those guys don't make an important amide hydrogen bond. They're actually sticking out, and they need to be capped. The second thing you always have to worry about with proline is steric interference. Phi-psi-wise, it achieves helical phi-psi just fine. Okay, but it can't supply the appropriate hydrogen bond. Same with beta. It can do beta just fine, but it can't supply the hydrogen bond. So, to make a long story short, it's actually not so easy to find these sites. We don't have steric interference. And so there are 2 examples that were published. Actually, this is one of the first papers, I believe, where anyone used single-point mutations to stabilize a protein. Okay, first, certainly ones where entropy was used. In an engineered fashion. People had identified stabilizing mutations before, but nobody had engineered them with this principle in mind. So changing a glycine and alanine gave 0.4 of a kcal per mole stabilization, did about half of what you expect. Allo to pro gave about 2/3 of the expected stabilization. So it does work, but the question you ask is why not as predicted? We've been able to calculate what we'd expect to see by increasing the number of states or decreasing the number of states available. But why don't we get the stabilization we expect? You could say, it's the theory that's wrong. That's fine, but there's a probably more obvious reason. Anyone got an idea? Yes?
Not to answer the question, but to go back to where were these residues?
They were in solvent-accessible regions that were in normal phi-psi angles and not making hydrogen bonds. Actually, sorry, take that. Gly-77 is in helix. Okay? It's in the long helix connecting 2 domains. Proline's at the end of that helix. But it's not still making any sort of hydrogen bonds. Try that out. So, any ideas? Well, you can look at a structure and say, I'm not going to cause any problems, but in fact, you pretty much often do. Okay? None of these sites are ever perfect. Okay? That's just how it is. In fact, it sort of makes you think that if this could have happened, it probably would have happened before easily. And so it's always, again, how much you can increase the free energy of the unfolded state without introducing strain or bad things into the folded state. And that's typically the explanation for why mutations rarely act as designed. As I said, you could certainly come up with other explanations. Okay. A better strategy, as it turns out, what nature has done, which is disulfides. Well, actually, I did want to bring one thing up. So this proline business is actually kind of interesting because there are a few weird bacteria that have a lot of proline-rich proteins, and they are thermophilic, and they live in the off the Sea of Japan. And they have proteins, some of which are 10 to 15% proline. And that's peculiar because proline is usually kind of a rare amino acid. So those guys obviously have an unusual metabolism where they're making an unusual amount of proline. But one of the things they're doing with it is creating super stable proteins. So at least evolutionarily, there's been one clade of organisms that have tried to do that. But most organisms use disulfides. Okay, it's, I suppose, maybe significant that those organisms live in an anoxic environment. So in a disulfide, you've got 2 cysteines that, when they're free, act just like any other residue, but when they're oxidized, now the residues between the 2 cysteines and the cross-link are not able to explore all the conformations. And you should memorize this equation. I'm joking. Okay. This equation describes for a freely jointed chain like a peptide how many conformations you lose, okay, and whatnot. And it turns out you can reduce this to something fairly straightforward, which you still don't need to memorize. But basically, the entropy reduction for a loop depends on how big the loop is. And the larger the loop, the greater the reduction entropy, but it scales as the log of the length, not as the length itself. Okay. So closing a reasonable sized loop gives you a lot of stabilization. You close a bigger loop, you get more, but you don't get it proportionally or linearly. It scales its log. So that's very nice. Okay, and so yes, indeed. Okay, when you reduce the protein, when you oxidize the protein from its reduced form to oxidized form to form the disulfide, you decrease the number of states, and that increases the free energy of the unfolded state, increasing the split between them. Okay. So if you were a protein engineer and somebody came to you and gave you a protein and said, hey, I want you to stabilize this for our new product, okay, what would you do? You'd say, well, let's go put in some disulfides. So where do you add these things? And this is the devil, okay? So you need to choose two residues that are nearby in the structure because they have to be able to connect together. They should be as far apart in the sequence as possible. Again, larger loop size, more stabilization. Even more so, they really have to be in a geometry that's going to be able to accommodate a disulfide. And that turns out to be best the case if you can find two residues whose what I call alanine core, out to C beta, more or less looks like a known disulfide geometry. And it turns out that this basically is almost never the case. Okay, it's almost never the case that you can find two residues that are pretty close to the disulfide geometry. And that's really why, while this does work, it doesn't work, again, quite as advertised. Most of these sites are rare, so you have to compromise. So some years ago, actually about 30, that's kind of scary, somebody came up, that's 30. Somebody came up with an idea to do this with T4-lysozyme. This is Matsuzuru Masazumi. And basically what he did is he found 3 different sites to put in disulfide bonds. He inserted them. And then measured the stability. Now, the question is, how do you measure disulfide stabilization? This sounds easy, but it's actually kind of a problem. You can use the usual methods, like urea denaturation or thermal melts. For urea denaturation and thermal melts, urea denaturation needs to be done in absolutely oxygen-free conditions for the folded form. And when you're, for the reduced form, and for the oxidized form, the problem is as soon as you open up the protein, disulfides shuffle. That you can suppress with pH, but then you're starting to get into pH issues. So it's actually somewhat difficult to measure reversibly, the melting behavior of these guys because the disulfide bonds themselves shuffle. So there's another way to do that, and you can make use of our good old friend linkage relationships. Okay? So you can measure the strength of the SS bond in the folded state and then calculate how much free energy you've lost. And so this is based on the same idea that we did with pKa's, with salt bridges. And the thermodynamic cycle goes like this. We have the folding of the reduced protein, which is kind of like what we're used to. You have the folding of the oxidized protein, and what we're interested in knowing is what's the energy difference between this and this. That's the disulfide stabilization, or not. And I don't think it takes a lot of math wizardry to know that I'm going to eventually get this difference to be equal to this difference, right? By the same old linkage relationship. I'm not going to go through that. You can do it yourself. I know a lot of you already did it to study for the exam, so you should be super experts at this. Okay. So the stability increase due to the disulfide is due to the difference in disulfide bonding energies. And I'm going to ask you, does this make sense? In the very same way that the changes in pK is linked to the changes in folding energies make sense, this is the same. So if we're talking here, about the stability increase due to the disulfide, what we're saying is really how much entropy was lost in the unfolded form of the protein when you form the disulfide, and that's due to the proximity of those guys, versus what's the energy difference between making a disulfide in the folded form and the unfolded form. So here the case is what folding is doing is you're losing a lot of entropy on unfolding. If you can pre-lose that entropy by bringing 2 parts of the chain close in proximity like they are in the folded form, you're closer to the folded form. That's one way of thinking about it. So that entropy has been lost already. The protein doesn't have to pay for that when it folds up. Likewise, for forming a disulfide or breaking a disulfide, the energy of that does involve covalent bonds, but whether you're in the folded or unfolded form, the covalent bonds are the same. They're not any different. Their energies are the same. But what's different about the free energies of forming those is how much entropy you have to pay to bring those guys together to form the disulfide. That's what's going to be the free energy difference between the forming a disulfide in the folded and unfolded form. And of course, I think you can see that it's the very same entropy that you lose from the unfolded state is also the entropy that you don't have to pay for to form that disulfide when the protein folds up. The protein folds up. It's paid with folding energy to bring those two residues close together so the disulfide can form. All right? And so obviously then that is going to lower the free energy of forming that disulfide. So what I'm saying is it's much easier to form a disulfide in the folded state of the protein than in the unfolded state because the rest of the protein is paid for the entropy. On the other hand, if you create a disulfide in a protein, you've pre-folded it to some extent. You brought two residues together that are otherwise not close together, like in the folded form. You don't have to pay as much entropy to fold. So it's two sides of the same equation. Does everyone understand that? That's true for all these linkage relationships. Okay, so what were the results? Well, you can certainly look at it in terms of melting, like here. These are, and I'll get to this in a second. Or you can look at disulfide energies here. And I'm just going to spend a little time telling you how, that's done. So, again, the problem with disulfides is they tend to shuffle, but you can suppress that by putting them in acid. When you put them in acid, they stay rock solid. And so what is done in this particular experiment is that you can add disulfide reagents to a protein to induce disulfide formation. You can change the redox potential by varying the amount of disulfide exchange reagents. And it turns out dithiothiritol, which is a reducing agent, will catalyze the formation of disulfides. Glutathione is another biological molecule that actually is very important in catalyzing disulfide formation inside of cells and maintaining redox equilibrium. So what you do is you change the redox potential in your solution by how much, in this case, DTT you add, or you can do it again by adding glutathione, and then you see how much oxidized form of the protein you get. You can do this in the unfolded and folded states, and you can get differences in the redox equilibria and calculate it that way. That's what this number comes from. Okay? This is the calculated version of that. We'll get back to this. So this is, and then how do you tell the oxidized and reduced apart? You can denature them in acid, put them on an HPLC, and they separate. So you can measure the peaks for the disulfide-containing one, and it's different, has different mobility than the one that's lacking in disulfide. So those can be integrated and quantified. And so that's what all this is about. So these are different mutants. So let's talk about this first. So there are actually 5 different positions that were chosen for disulfides. And what these are melting temperatures of the reduced and oxidized proteins. I said it wasn't very easy to do this, but it could be done. It just wasn't very, and not very satisfying, not particularly reversible. So, if you believe it, basically you see the reduced forms of the proteins all have lower stabilities than their oxidized forms. So the disulfides are behaving themselves. They're doing, in a sense, what was expected. However, this is the temperature of melting of the wild-type protein. 2 of these mutants were pretty destabilized by making those cysteine substitutions, and the disulfides did not allow them to recover a stability that was greater, than the wild-type protein. So these did not work, in a sense, for stabilization. These, on the other hand, worked rather well. This protein here has 12 degrees increased TM. That's pretty stable. So the fact that all of these disulfide mutants are less stable in wild-type tells you that you're messing with the folded form of the protein. You're not making it very happy, creating cavities, steric clashes, and so forth to put these in. But then when you close the disulfides, those proteins are more stable than their parent protein. Yes?
So it's not necessarily an absolute that a large loop size is always going to have more stability.
No, because why? There's other things. It turns out that these two are about the same, about the same stability change, and yet the loop is a little bit smaller. Now, when you start taking logs of these guys, you can check it out. It actually comes fairly close to prediction in terms of the relative change, But no, because why? Because you can introduce strain into the folded state, or bad things, Okay. Yeah?
Did they try adding 2 disulfides? I know.
Oh, yes, we'll get to that. Very good. Good question. Okay, so this is the calculated, this is the expectation for disulfide stabilization. These are the real numbers, of what actually happened. And I just want to show you, over here, this protein, for example, becomes reduced much more easily at much lower DTT concentrations than these proteins. This is the bad disulfide mutants, and these are the good ones. And so what you can see is in some cases, like these here, they're really, really pretty far off what's predicted. And even so, all in all, you're lucky to achieve half of that. Some proteins are less stable than wild type. Why? Because the mutations do bad things. And then when you oxidize them, it can get even worse. So this part, I think we can predict pretty well, although, in fact, I think that pretty much corresponded to the theory. But this is the part that does not do well. Why? We can introduce bad things, which I'm calling strain, by just making the mutations. And then when we oxidize it, we can even cause more problems, right? You can imagine if I have a disulfide, or I have two cysteines, they're pointed like this, and I have to rack them around to make the disulfide. Yes, I can make the disulfide, but that's going to be very strain-inducing. Okay. What's interesting about these guys is that you can put them together, and they're pretty much additive. That doesn't really fit too well into the theory, but that's how it is. So here's the situation. We've got 3 of the stabilizing mutants that I just showed you combined into 3 different double combinations. And shown in the dotted line, okay, is the expected changes, and shown in the bold line is the actual changes. Okay? So in other words, essentially, the expected changes from additivity are very close to the actual changes that we're seeing. Okay? And that's pretty remarkable. So, disulfides are additive. Yes?
Can you explain how to read that back?
Yes, I certainly will. So this is the TM of the reduced forms of the proteins here. This is the wild-type TM at 0. Okay? These changes in TM, pardon me. Okay? And then these are the TMs of the oxidized versions. So this gap here is the difference in TM. Okay? So these are the multiple ones that have bigger gaps because they should. And dotted line is just what's predicted by simple addition. Okay? All right, great. So, pretty cool. So I think to summarize all this, disulfide bridges are a great way to confer stability, and nature has done this, okay, quite a bit. Almost all extracellular secreted proteins in your blood have disulfides. They're heavily disulfide linked. Many hormones are heavily disulfide linked. TGF-beta is one of my favorites. It has a ring of disulfides, and then through the middle is another one, a 4th one. So that's pretty amazing. Yes, sir?
For the evolution, there may be disulfide bonds
sorry.
between...
Sorry, I lost my page. I'm listening to you, though.
Yeah, between, let's say, if you look at the first double mutant, there where it's 3 and 97.
Yes.
And then there's 9 and 164.
Yes.
But then, isn't there a freedom of also linkage between 3 and 164 and 9 and 97?
No, those aren't placed where they can do that.
No.
No, because they're pointing in completely different directions. I mean, if you made that, you probably would not be able to fold the protein up. They're not. I should say, disulfide would not be compatible with the folded form. And that's one of the problems, okay, when you melt these guys, is that they will shuffle like that, and then you get a mixture of different disulfide species, and so it's pretty lousy. Okay. Does that answer your question? Okay. All right. So, as I said, a large majority of secreted proteins are heavily disulfide linked for stabilization purposes. Okay, and it's additive, so more is better, and that makes it easy from an evolutionary standpoint to increase stability. The smaller amounts are gained by doing single mutations, Gly to X and Allo to Pro. And again, this is one of those things where if the protein needs to have more stability and it can gain it by doing that, it probably has. Evolutionarily speaking, these have probably been done. There are polyproline-containing proteins. I mentioned the ones from the thermophilic bacteria, but your mouth is full of polyproline proteins too, and they're quite stable. Your salivary proteins have lots of proteins in them. So one of the things, the lesson that comes out of all of this is that design mutations can be successful. I mean, we do understand enough to do this. But you definitely get less than expected stability often. And really the explanation for that is that the effects on the folded state aren't easily predictable. And particularly if you're talking about relatively small free energy differences between the mutant and the wild type. The mutant and stabilized protein. Wild type and stabilized mutant protein. I'm having trouble today. Then any sort of calculations you do will be in huge error. And so this is really where I have to say that programs that try and calculate stability of proteins really don't do a good job with very subtle changes in stability. It's just not so good. So and really the problem is the strain. There's problems that are caused by the mutations, or if you're making a disulfide bridge, there's an additional problem where to accommodate the geometry of the disulfide, you might have to do something not so good to the folded state of the protein. Okay, whereas in nature, I can tell you that that's not the case. That basically those appear to be pretty much unstrained disulfides. And that's because the rest of the structure has essentially evolved around that. You can imagine an initial event where a disulfide is created kind of by chance, it's stabilized, and then the rest of the structure is adapted by mutation to make that disulfide even better. Okay, so that is mutational analysis. Are there any questions? All good with that? Okay, expect that on the final. All right. So, sorry, I'm a little behind here. Should have queued up the other one.
What was the protein that had the ring structure, the ring of disulfide?
TGF beta. And that fold is actually common amongst a bunch of different hormones, and I think cytokines too. I'm not sure. I think one of the interferons has a TGF beta. One of the ILs has a TGF beta like fold. I just don't remember which. Kind of not my thing. Okay. Okay, so now we're going on to the last bit about structure. So, the real, I think, value of this class, if nothing else, is to understand on some sort of intuitive level how sequence and structure are determined. And actually, from your from your exam, there's one question I asked about this, and it seemed like most of you had gotten it. Maybe you've seen a previous exam that had that question, and you pilfered it, and that's fine. But that's really the basis of it. So what we really want to understand is, what's the information contained within a protein sequence that makes it the shape that it has? Taking that knowledge, how probable is it for that kind of structure of any sort to arise from a random pool of sequences? How guided has evolution been to give us these structures? And in the end, do we know enough to be able to design a protein fold? So I'm going to tell you about a series of experiments. I'm going to go through them a little more quickly than usual because of time constraints, but I'm not going to leave out any of the important details. There's just a lot of little asides. So one of the first experiments that really was designed to probe this kind of question, and it's similar to an experiment I told you about that I had done as a postdoc looking at protein cores. This one was a little bit more open-ended. And really the question is, what's the information contained in the protein sequence that specifies the fold? And this is from a Science paper in 1988. And so what these guys did is they took a protein that they were interested in. As a small protein, it's something called lambda repressor protein. And lambda repressor protein's job in the world is to bind to a piece of DNA. And we'll get to that in a second. And so what they did is they essentially went through the protein. They engineered a gene, which in those days was sort of a big deal. And the gene had lots of restriction sites. Sorry about the resolution of this. And how over time this has gotten completely corrupted, and I don't really know why. But what this is supposed to show is a bunch of codons for amino acids, which are shown up here. And what they did is they chose one or two or three amino acids in a contiguous stretch. And they basically allowed those positions to be anything. Not just one amino acid, but they created libraries of proteins where at those positions there were any number of possibilities. For example, if they have two sites, there are a total of 400 different combinations of proteins that you could create. And the way that they did that is they used synthetic DNA to do mutagenesis. And in those synthetic DNAs, the codons they wanted to mutate, they made codons that were XX, any base, and then G or C. You could do XXX. There's 64 codons. There's a problem with that, though, because the redundancy of the code, some amino acids like leucine and serine have 6 codons for them, and some of them, like tryptophan, have only 1. And so you have a huge bias against the low abundance or low codon amino acids, and plus you have 3 stop codons, which means you have 3 out of 64 chance of generating a non-protein. And so to kind of get around that, you can reduce the last position to be either G or C. That brings you down to 32 possible codons. Now the biggest bias is 3 to 1, and there's only 1 stop. So that's one solution. Nowadays, you can be really fancy, spend some money on DNA, and get exactly a 1-to-1 mix of all the possible codons, but that's not what they did then. So they made these little cassettes, little synthetic duplexes. They had restriction sites designed to their protein, and they just dropped these cassettes into them and then transformed it into bacteria to generate their libraries. And when they have that, now they have a bunch of colonies on an E. coli plate that basically have different lambda repressor molecules being expressed in them, because these are on expression vectors. So what does lambda repressor do? So phage lambda is a bacteriophage. And it has a lifestyle that's quite interesting, one of the first bacteriophages that was really dissected genetically. And what lambda does is it lands on the surface of a E. coli cell and injects a piece of DNA. And that DNA then goes to start to make virion particles. The cells fill up with the virion particles. They have a lysozyme that busts them out, and they do it again. That's called the lytic cycle. So they form, they kill cells basically by filling them up with bacteriophage particles and then bursting them open. Kind of like T4 phage, as I said before. That's called the lytic cycle. But what Lambda has, which is actually a really great innovation, is that sometimes instead of making more of itself, it dives into the genome, okay, by recombining at specific sites in the bacterial chromosome, and then it shuts itself off and just sits there. So how does it shut itself off? Well, lambda repressor is the protein that basically shuts off the entire virus. So it just sits there quietly. And it makes a little bit of lambda repressor that keeps the bacteriophage happy, and all is good. However, at some point, occasionally bacteria get upset. They have DNA damage, and that activates something called the SOS system. It turns out that the recombination protein REC-A binds the lambda repressor and causes it to autocleave, to inactivate itself. And that, in turn, turns on the lytic program for the phage, and then they kill the cells. But as long as that cell is carrying a prophage, an integrated phage that's been quiet, it cannot be infected by other phages, because as soon as they come in, the lambda repressor lands on its DNA, shuts it off, and then eventually those get killed or diluted out by the bacteria. So, lambda repressor confers resistance to lambda phages. Okay? So if I take my plate of bacterial colonies and I plate them onto a plate that has a bunch of lambda phage in the agar, they will die unless they carry a functional repressor. Okay? So that's good. So when there's no phage, you get the same pattern of colonies you got before, but when you have phage in the plate, only the ones that make a functional protein will live. So that's pretty clever. And so what they're able to do then is find the survivors, but they also could classify the non-survivors, okay, by the ones that plated on without phage but wouldn't plate with phage, because these are replica plates, which means they have the same pattern of colonies on both of them. So that's pretty cool. And so what they're able to do then is then take those and sequence them and generate large banks of sequences where they have at different positions, at different stretches along the protein, different amino acids. And from that, they built up a set of tables, okay, which I'm just showing for one stretch of amino acids, that shows what's the tolerance of a particular position to substitution. If you are allowed to have more amino acids at a particular position, that's a tolerant site. And if you aren't allowed to have very many amino acids at that position and still be functional, then that's not a very tolerant site. And shown here is kind of this typical bit of data that they have. So in the white bars, that's showing percentage of amino acids that were allowed at that position. Some of them allowed, up to 14 out of 20 amino acids were allowed. In other positions, maybe 1. Okay, so that's not that's quite different. What they did then is they showed that the tolerance mapped with essentially the accessibility of those sites to solvent. What I mean is that the surface positions were highly tolerant to mutation, whereas the interior positions, not so much. That sounds a lot like what we already knew. Okay? But this is a sort of large-scale, kind of early pre-omics approach to doing that. So this is another look at the data. This is just an expanded view. This is a long stretch of a particular set of positions. This is, again, the fractional accessibility. This is a solvent-exposed site. This is a completely buried site. And above here are shown what amino acids are allowed at those positions. And they line up pretty much as I'm saying. Okay, now, there are several different types of sites here. There are sites that are highly tolerant to mutation, okay, where pretty much almost everything's allowed. And more or less, those track at the surface exposed positions. There certainly are exceptions like this one, okay, and maybe you could say this one, but these are pretty big. The second, okay, are ones that primarily are hydrophobic. And probably, honestly, I should have put this one in that bin, okay. And there are fewer of those, so the majority of the sites actually were highly tolerant. A few that are constrained to be primarily hydrophobic, and then there are ones that just have to be what they are, regardless of their surface exposure. So, for example, here's a pretty exposed site that has to be proline. I think we probably figure out what's going on there. Proline's a special amino acid. But this one's got to be isoleucine. And what that means is that if you don't have an isoleucine there, this protein doesn't work. And it turns out that guy is right at the DNA binding interface. Okay, it's actually making sequence-specific contact with a thymine residue. So, if you go in the protein interior, just like what we had shown, you essentially have a lot of amino acids. This is showing the interior of the protein. It's a little mini protein core. And these are the substitutions allowed. Now, these are substitutions done one at a time, not multiply. Okay? And so what you can see is that there's a large tolerance at the interior, but the one thing that seems to be true is the predominance of hydrophobic amino acids at these positions. Again, this is reinforcing what we already know. So the surface... this is just another way of showing it with a picture. So shown here is DNA. This is lambda repressor bound to DNA. And these red balls, okay, are sized by how tolerant they are to amino acids. And basically, this is just showing that essentially the tolerant sites are on the surface of the protein. You take away the protein, you just leave the sites. You can see it looks roughly the same, which is just simply telling us that, yes, we're really looking at most of the tolerant amino acids not hidden away. On the other hand, if you look at the sites that are constrained to be hydrophobic, you don't really see them when you show all the amino acids, but when you strip everything away, you see the hydrophobic core of the protein lit up. Okay? So, again, hydrophobicity seems to be the most important part of this. What's interesting now is then you can take this and you can apply this to kind of looking at a bunch of proteins that are in the same, same structural family, or orthologous proteins. So it turns out Antennapedia is a particular type of so-called helix-turn-helix protein, which is a type of DNA binding motif. They're actually called homeodomain proteins, to be precise. And this is a hydrophobicity plot of that. And the way these hydrophobicity plots work is you take the amino acids within a certain window, you average their hydrophobicity values, or sum them up, however you want to do them, and then you plot them, moving that window one residue at a time down through the whole thing. And what you can see is you can see some very pronounced spikes of hydrophobicity in the sequence. Why do you use 8? You can go talk to them about it. Okay. On the other hand now, this is Antennapedia, so it's a prototypical homeodomain. If you then take 39 homeodomain proteins, okay, and average their profiles together, hydrophobicity profiles together, you see very clearly 3 big spikes of hydrophobicity. So what this is saying is that these hydrophobicity spikes are actually predictive of the fold of a homeodomain. And in fact, are predictive. They have predictive value. So the take-home from this experiment is 2, that there are essentially different types of residues that have different sensitivities to substitution, and they have a lot to do with where they are in the protein. Essentially, 70% from this experiment, 70% of these sites could be essentially anything. They're solvent accessible. Those are the solvent accessible sites. And this, again, goes along with our understanding of the mutational analysis, where we know that if you mutate surface sites from a stability standpoint, you don't really cause much change in stability. About 20 to 25% are hydrophobic. Those are the buried residues, and that's about the fraction of residues that are buried in most proteins. And that amount of hydrophobicity is typical for a soluble protein. And then 5 to 10% of these guys have to be exactly something. And these are the ones where the protein just simply will not work unless those residues are those. That's 1/10 of the residues, or less, or 1/20 of the residues. So most of the residues are expendable. That should have been what you'd seen if you'd been able to get a good set of sequence alignments, divergent enough set of sequences in your MAXI project. That should have been what you saw as well, that there were really very few highly conserved sites, and almost all of those were located in the functional regions of the protein. Glycines also are conserved because of fold. So essentially, hydrophobicity patterns are shared between homologs, but they're only constrained to be hydrophobic. So this was kind of annoying to people because it didn't really sit with the idea of specificity. We look at these protein cores; they fit together so beautifully, and yet they can be anything that's hydrophobic. So telling us that really all how that fits together is a detail. So the other thing that comes out of it, which is more exciting, is that maybe that's all that matters. Maybe all that matters is you simply have to bury certain sets of residues, okay? And that the constraint to bury the hydrophobic residues is a huge constraint on the ability to form a fold. And that is the take-home message. That essentially, and I'm saying this with emphasis for a reason, okay, that essentially the constraint of burying a certain pattern of hydrophobic residues greatly restricts the number of structures that can do that. And it's the single biggest, I'm just going to give you the take-home, it's the single biggest reason that proteins fold up the way they do. But you have to have proof. So that comes next. It's not perfect proof, messy a bit, but that's good because then we can talk about some real science. Because real science is messy. If you ever hear a story that sounds really pat and clean, it's probably a lie. Okay, or there's probably things being hidden. It doesn't mean it's wrong, it just means that it's maybe not quite honest. This was honest. Okay, so the question is the hydrophobicity pattern enough to make a protein fold in a particular way? This is a very famous paper by Michael Hecht. Didn't get tenure, honestly, from this, which is shocking, but oh well. And his hypothesis was that all it's required to specify a protein fold is the correct pattern of hydrophobicity. And so he liked to work with this protein called cytochrome B562. It's a 4-helix bundle, which hopefully you can see. Normally it binds to heme, like if you express the wild-type protein in an E. coli turn red because they grab a bunch of heme from the E. coli and store it up. What he did is he looked at the crystal structure of this guy, and this is just kind of a pictorial view of the crystal structure, and just simply identified the buried positions, okay, all the ones that were solvent accessible. And you can do that using various programs. And then the ones that were exposed to solvent, those are another position. And what he did is he sort of wiped out their identities and said, I'm going to call all the ones that are buried H's, and I'm going to call all the ones that are exposed P's, and I'm going to make a whole lot of proteins with that pattern to see if they fold up. The big swindle here, okay, is that the loop positions were kept as the same as cytochrome B562. That's the swindle, okay? And that was because in an earlier paper he had shown that he could mutate them in the context of the wild-type protein, and it didn't really matter what they were, okay? That's, as I said, a swindle because it's not, then he should have just randomized them completely. But anyway, all right, so he didn't do that. So anyway, how did he do this? How did he do the experiment technically? So what he was able to do, again using this clever business of random codons, is create a library that just simply had amino acids not specified by their identity, but rather by their character. And luckily, the genetic code makes this really easy. If you take any codon with a T in the middle of it, it will be hydrophobic. Okay? It will either be leucine, methionine, phenylalanine, valine, or isoleucine. Okay? That's the truth. So nature has already segregated these for us. And so it's a simple matter to make a random codon where you're allowed to have all 4 bases in the first position, have only a T in the second, and then G or C in the third. Likewise, to make something polar, if it has an A in the beginning and it starts with A, C, or G, the reason they use T is gives you 2 stop codons, okay? And A, C, or G in the last, you get all polar amino acids, lysine, glutamine, glutamate, asparagine, aspartic acid, and histidine. And so he just made genes with the appropriate pattern of hydrophobic and hydrophilic amino acids and then expressed them in E. coli. And then he had to identify which ones were folded. Now, I happen to know that he is a very optimistic guy, and what he was hoping is that he'd see red colonies, where they've took in the heme and did the whole thing. But that's absurd, okay? What a ridiculous thing. But anyway, we can be optimistic. But now, the problem is, if I were you, okay, so you do all this and you don't see any red colonies, do you stop? No, you say, well, I'm looking for folded proteins. Let me ask you in sort of a semi-high throughput way, which now, in today's terms looks silly, okay? But at that time, it was a bit of work. There weren't very many graduate students, no robotics or anything like that. How would you identify folded proteins that have no activity? How would you, if I give you an E. coli colony, what would you do to demonstrate that there was a soluble protein being made inside of that? Yeah. Well, you'd have to purify it, wouldn't you? Okay, so yes, you could. Yeah, they said by day's terms, sure, I could purify 100 proteins in the afternoon, but then nobody would have thought of doing that. So in an unpurified, sort of semi-high throughput manner, that you could get a graduate student to do without quitting. So what are the attributes of folded proteins? What's actually really the first attribute for the most part, the majority of them? Pardon me? They're compacted. Okay, but how would you assay for that?
Could you break up, use something to break up the proteins?
You can say if they're protease resistant. You could do that, but why would you need to do that when they're already inside of a cell? You got in there though.
Can you run a gel?
On what?
Like a Western gel and then use an antibody? And
You could, but what would you do?
Would run faster?
Well, okay, so explain how you would get the sample to put on the gel.
You slice yourself
Okay, then what?
and run.
Yeah, so you run this. So would you add SDS to the buffer? Okay, and the problem is that you have all these charge residues that are all in different charge states. So yes, you could kind of do that and run an isoelectric gel. They didn't do that, actually. They didn't run a native gel. Okay, but did something which technically was probably a lot less challenging. Close though, you guys are getting it. So let me ask you, if it didn't fold, what would you expect in your experiment?
My folded protein would run more slowly.
Okay, it's an interesting idea. All right. Any other ideas? You're nibbling at the edges of it. This is going to sound so silly when I tell you. So how do you identify folded proteins? Okay, the first thing is that folded proteins are compacted, and therefore they're protected against proteolysis. So that was kind of on the right track. What else, though, that's more kind of fundamental? If a protein is folded, okay, let's put it this way. When a protein is not folded, what happens to it? Remember fried eggs? They aggregate. And inside of E. coli, when things aggregate, they're shuttled off into these little things called inclusion bodies. So, in fact, folded proteins in E. coli are soluble. They're in the soluble fraction. And they're also protected against proteolysis. An unfolded protein in E. coli, most times, I'm not saying always, most times will be degraded. So, they did something very simple. They grew their cells up, they lysed them, and took the soluble fraction and made a gel, like you said. Except there's a denaturing gel. And this protein is small, so it runs quickly. These are really kind of ugly gels, but there they are. There's the proteins, okay? And when they did this, they found that 29 out of 48 sequences that they looked at. Nowadays, you just get laughed out of the house for doing 48, but then that was a bit of a deal. 60% of them made a protein that was both soluble and not degraded. 60%. That's a lot. They then sequenced these guys to make sure that they really did have the correct amino acid pattern. In fact, they certainly did. However, they weren't able to tell the ones that were soluble and not soluble, because they also sequenced those from each other. There wasn't anything obvious that made the one soluble. So 60% of them. So the real question is, well, are these really folded? So again, now that you have purified proteins, so they purified some of them, a few of them, 3 actually, what would you do now to determine whether they're folded? Okay, now you have purified them. Now we can start where you were beginning before. No idea? You even said it before. Yeah.
Try to chop them up.
You could try and chop them up. They didn't do that. Okay. They did something that was a little bit better in terms of at least establishing whether it's size. Because, chopping up also has to do with amino acid composition, because proteases are non-specific.
Run a gel out
They already did that. Okay. Somebody mentioned it. Yeah.
Try to detect the helix.
Yeah, how would you do that?
Polar light mixer.
Yeah, CD, exactly. Okay, so CD, that's what they did. So they did a circular dichromosome, okay, and these are basically 2 mutant proteins and the wild-type protein, and they have CD, they have helical CD, as expected. A little bit less on a per milligram basis, but to be honest with you, they're probably not well quantified. They're probably not that pure. So they did have helical structure. Okay, that's the one thing. What's the second thing that you might be concerned about? If they're just because they're helical, does that mean they're folded into the structure that you thought they were? And what alternate structures might they form? What about their size? Right? If they're monomers, or the size of an expected monomer, then that means they're probably folded. Because remember, those methods to determine molecular size are calibrated with standards. So are they monomeric? What do you use to determine that? How do we determine molecular size easily? Gel filtration. You can also use light scattering and all sorts of stuff. Again, this was before that, believe it. So gel filtration. So when they did that, they actually got a gel filtration peak that was pretty much exactly dead on the molecular weight. That suggests that it's forming a monomeric structure that has the approximate molecular dimensions that they thought. Okay? The one thing, though, that I would have noticed, if I were the referee of this, I wasn't, was that there's this kind of big shoulder here, which means maybe they're aggregating a bit. But fair enough, okay? They're monomeric. Yes. Are they stable? Well, then we do melts. So this is a urea melt. And this is a very nice urea melt. You get delta Gs, and you get a lousy delta G of 3.5 to 4.5 kcals per mole for these 2 ones that they did. But hey, they're folded, and there's a folding transition. I'm going to say sort of, because this is a very long folding transition. Okay, usually these occur over, at best, 2 molar change in urea, and this is occurring over something like 4. So this is the first guess that this isn't quite really a normal folded protein. Now there is a gold standard, okay, for folding. I mentioned it a while ago when we were talking about some structural techniques. Does anyone know what the real gold standard, besides crystallography, okay, is for a folded protein. Anyone remember? It's NMR. Okay, so in NMR, when a protein folds up, you take a bunch of residues that are all in the same environment, remember that? Okay, where their resonances are all piled on top of each other and they're really not distinguishable. And then when they fold up, now all these resonances have different environments, and that spreads out to chemical shifts. That's called chemical shift dispersion. Okay? And basically, the NMR of this guy shows no chemical shift dispersion. Okay? And that suggests that it is not uniquely ordered. It doesn't mean it's not folded, I mean in the sense that it's not folded up, but it is not a real protein. It's a wiggly thing. Okay? Which we in the business call molten globules. So, it's definitely not uniquely ordered. So, the take-home for this is that certainly the pattern of hydrophobicity specified a compact structure. The caveat, of course, is that the terms weren't random, so maybe the terms help things a little bit. Those patterns of hydrophobicity ensured solubility and monomericity. They likely had the right secondary structure, but pretty poorly ordered tertiary structures. And so the take-home, at least from my point of view, is that the hydrophobicity pattern pretty much probably specifies the fold, but it doesn't do enough to make a real protein. There has to be something more than that. And so the question I'm going to ask you now is, how is, given that you can get to a helical bundle structure but not all the way, how could you stabilize a particular structure over other ones? So when you say things aren't uniquely ordered, what that means is that you have a structure that's either fluctuating a lot or folds into a bunch of similar but not the same structures in a dynamic fashion. So let's go to our conformational free energy diagrams, which I really like. So this is a diagram showing basically on this axis conformation, which is hugely multidimensional thing. But now I've just been hearing about all the genomics people now collapse 1,000 dimensions down to 1 to make Tisney plots, so we can do this too. All right, so we have a conformational axis here, and all it means is anything close by on this axis is similar in structure and far away is not. If we look at a peptide that never folds, what we have is we have an energy surface that's bumpy, because obviously different conformations have different energies, but it's not so bumpy that any one of these structures is favored. Basically, there's a bunch of structures that this thing can take on, and it can dynamically interconvert between this. This is a peptide that never folds. It's always unfolded. Okay? On the other hand, naturally occurring proteins have a different situation. They have one structure, or one set of structures down at the bottom of this thing, that's much lower energy than everybody else. And structures nearby are of a lower energy, okay, but they're not that much lower. There's something that happens when you get very, very close to the final structure that locks in a lot of free energy. Okay? And so in this sort of thinking, we have unfolded states out here, we have folded state at the bottom here, and then these kind of partially folded structures, ones that have bits and pieces of the folded structure, are what we call folding intermediates, or partially folded intermediates. Okay? And another picture you might have seen from some of the reading is something like this. It's a two-dimensional surface. And here's the folded structure down at the bottom. This is the unfolded states. And as we get closer, there are these little bumps that are folding intermediates. Another picture you might see in the reading is something like this. And again, the thing to remember, and I think this is important, as you go down this so-called folding funnel, so again, out here are all the unfolded states. As soon as you enter this, you're getting closer to the folded structure. But as you go down here, you're acquiring more and more native structure. And this has been well demonstrated. I used to talk about this a lot when I first started lecturing, but this is so well demonstrated we don't even have to talk about it, that basically when proteins are go on their way to folding, they're acquiring more and more native structure. They're not doing anything unusual and then coming all of a sudden into the native structure. And actually that was one of the messages of protein folding in vitro and in vivo that I can still convey to you, that folding intermediates are just partially folded native structures. There's nothing weird or different about those intermediates. Okay. So that's the situation with native proteins. Now what about our design proteins? Well, they still have unfolded states, but now they have lower energy, what we're calling folded states, but there's a lot of them. They're around the same structure, okay? They all have the same residues buried mostly and so forth, but they're not really sitting in one particular structure. There's a number of isoenergetic structures that it just flops between. And that's not enough to give you NMR shift dispersion. In fact, you might think of that as really that those guys are really kind of intermediates on the way to the final folded state, but because you don't have a molecule that can get there, it doesn't. So the question is, how do you stabilize your structure, the actual real structure, away from all of these. So somehow you have to destabilize all the structures that aren't the native form, and you have to singularly stabilize the one that is. So what are the structural elements that are responsible for stabilizing a particular conformation? Any ideas? How do we get this? And what would you need to do that? I think is the question. What would you need to do that? We already know that hydrophobicity patterns definitely don't do that. Right? What sort of energetics do you need to stabilize a particular structure very precisely, okay, but destabilize all the other ones?
So you change the environment.
Okay. Do you want to be more specific?
Well, we know that the environment can't favor. Like if you change the environment.
Oh, you mean like the external environment?
Mm-hmm.
Yeah, well, I'm talking about in water, though. Nothing fancy. Okay, so I'm talking about from a sequence point of view or from a structural point of view. What structural elements would do this? Pardon?
Using things we just talked about, like hydrogen bonds and ionic interactions.
Okay, so let's go through it. Any other one? Any other ideas?
So for the vandals, like maximizing your.
Okay. All right. So hydrophobicity patterns. So yes. Okay. So van der Waals interactions. Okay, they're not directional. So hydrophobicity is just I'm burying this stuff. I have to sequester it from water, but it's easy to do that. Okay, I just have to bring these things together. And it turns out because of the helical patterning of those residues, you form alpha helices and they kind of come together. That excludes water, but that's all hydrophobicity cares about. There's lots of different positions that exclude water. On the other hand, van der Waals interactions, we think about lock and key fit of residues, right? And now I have two residues that are fitting together very nicely. If I move them just a little bit from that position, now what have I done? I've created unfilled space. I've lost van der Waals interactions, and I've probably also disturbed the interactions of a whole mess of other residues. So in fact, that lock and key fit that we talked about is important for achieving singular energetics, a single conformation's precise flowing free energy. But it's not enough because we don't see proteins that are just simply held together by that. So van der Waals interactions, absolutely, the lock and key fit the interior residues does change the free energy profile to deepen some and raise others. But the real one is these buried polar interactions. A buried polar interaction, when it's not satisfied, we know that that's highly destabilizing. We just went through a bunch of mutational analysis to talk about that. And yet, when you now put it into a productive hydrogen bonding situation, okay, now you get really good stabilization. So in fact, I'm going to argue that really the polar directional interactions, okay, that involve a number of residues, the more residues involved, the more singularly they will stabilize one form over all others. And if it moves just a little bit away from that, now you break a hydrogen bond. Now you've got two barium charged residues, or two barium polar residues. That's hugely destabilizing. So it pushes up the energy of the conformations that are close that don't make those interactions, and it really lowers the energy of the ones that do make those interactions. And again, the more of those that you have involved, the fewer number of possibilities that will satisfy it. So that's the answer. So van der Waals and barium polar interactions stabilize the individual specific forms. If you think I'm going to ask a question about this in the final, you bet. Okay? So you're warned. So not only do these stabilize specific conformations, they destabilize the nearby ones. That's how we, basically take these guys and shove them up and take this one, push it down. Yes? So there were two hands. The three hands, okay.
What differentiates the hydrophobicity pattern from the barium polar interaction? What gives it the directionality?
Okay, so hydrophobicity is just simply pulling things out of water. Oh, yeah. Thank you. You reminded me of a pet peeve. I'm going to take a slight digression. I saw the term hydrophobic interactions on your final. If you put it on, on your midterm, if you put that on the final, it would take 10 points off. Okay, there's no such thing as a hydrophobic interaction. I told you that early on, and I'm going to tell you again. If you're coming to class, you know that does not exist. I don't want to see you using it ever again in class, at least in my class. Okay? There's no such thing. It's a completely bullshit way of explaining hydrophobicity. Pardon my term. Okay, now it's on tape. You have any trouble. But it is. Okay? And I'll argue anyone that one to death. Okay? Because it's really just a disservice. There's no such thing as a hydrophobic interaction. Hydrophobicity is the exclusion of nonpolar surfaces from water. It's not a force, it's a real tendency. Okay? But when they're in contact with each other, they do better. That's it. Okay. So I wasn't yelling at you. Okay. But back to your case. The reason that hydrophobicity can't do it is it's not a force. Okay? It's just simply that all I need to do is get that stuff away from the water, and I'm good. The question is, how many possible ways can I do that? It's certainly way fewer possibilities than the protein exploring all of its conformational space. So it does narrow down the number of conformations hugely. Okay? But you still have a lot of leeway in how you fit those together. But now, if you put textured surfaces that fit together nicely, right, that's going to lower the free energy of only the ones that fit nicely together. Again, you move those away from each other. You create cavities and expand. Does that make sense? Did you answer the question? Yeah.
Would you say that the manifold interactions, I guess hydrophobicity, pushes it in the direction of more stable?
Variables, yeah.
First, then
No, hydrophobicity is first, right? So again, unfortunately, we won't get a chance to talk about the folding process, so I'll just give you a picture. If I take an unfolded protein, say an 8 mole urea, I throw it into water, dilute out the urea, so now it's unfolded. The first thing that happens is it bunches up. That's the hydrophobic effect. Now, once it's in this thing, okay, where the hydrophobic residues are hidden in water, then it wiggles around, okay, to fit all those pieces together. And usually the way that happens is that you've got bits of structures, so maybe you have 2 helices come together, and then other ones layer on top of them. And then at the very end, this is known from folding experiments, the polar interaction lock-in. They're the last thing, salt bridges are the last thing to lock in. So there's your hierarchy. So it's both temporal and energetic. So again, you get to a lower free energy structure by hiding the hydrophobic residue from water, but then to get the lowest energy structure, you have to have a precise fit. Okay. Any other questions? Okay, those are great questions. Sorry about the rant about hydrophobic interaction, but okay. All right, so now we're kind of getting into different realms. Now we're talking about evolution. So now we already understand that patterns of hydrophobicity are important, and we understand that the hydrophobic residues are really the singularly most important thing most important factor that determines the fold of the protein. Again, because there's very few ways that you can vary all those residues, maximize their burial, and still agree with protein stereochemistry and hydrogen bonding. Okay. But now the question is, all right, so from an evolutionary standpoint, how did this all happen? How hard is it to come up with a sequence that forms some sort of structure? So we're taking a step back and not talking about a specific structure, but just any structure at all. Okay? And so from random sequences, how often do you expect a protein structure? So one thing to do is just make a bunch of random sequences. And then screen for solubility, just like Michael Heck did. Nowadays, you could actually do this, but for some reason, nobody's taken it up to do it. They've done things that are similar. Maybe because there's really nothing to be gained. Okay, but essentially, if you make a completely random adiamer, you have a big problem, which is that there are way too many proteins to even think about screening. And so the success of that would really depend on how often you would actually expect this to happen. 10 to 100 proteins is 10 to 100 is a number that's so big that there's nothing in the universe that even approaches that size. If you fill the entire known universe with protons, you're talking about a 10 to 40th. Okay, so that's 60 order of magnitude different. So you can see this is it's not even reasonable. So that would be nice to use all 20 amino acids, but it's not reasonable. On the other hand, if you kind of take the evolutionary steps backwards, did we start with 20 amino acids? No, of course not. Okay, we started with some subset. We don't know what subset there was. Okay, we could probably guess. But maybe you could see what happens if you just use 3 amino acids instead of 20. That would make your statistics a lot better. Still, 3 to the 80th is a ridiculously large number. Now it's at least all the protons in the universe, though. Okay, so filling the universe. So basically, to see this, okay, the question is, how well could you sample this? And for people who do random mutagenesis type of experiments or library experiments, a millionfold is really, it's not the largest. You can do now a billion, maybe a billion, okay? And with RNA, maybe even 10 to the 12th molecules you can screen. But for the most part, you're not likely to screen a large fraction of this. So at best, if you can get 1 in 10 to the, you can screen maybe 1 in 10 to the 34 sequences. But again, how often do you expect these to occur? So somebody's got to do the first experiment, publish it, so what the heck, all right? You basically say, I'm going to make some random proteins and see if they fold. So Bob Sauer's group, once again, they've done lots of great work in this area, made a set of proteins called QLR proteins. And the bottom line is that when they made adiamer proteins composed of just glutamine, leucine, and arginine, 5% of them were soluble. Okay, so what's a QLR protein? It's a protein that contains glutamine, leucine, and arginine. Why? You have a polar residue, you have a hydrophobic one, and you have a charged one. Okay? The real reason is that they can make a random codon that would encode all three of those amino acids. And then they made libraries. Now, one of the problems if you did this in even amounts is that 33% leucine, which is with a T in the middle, is actually more hydrophobic than a normal protein. And when they made their initial library, they happened to get a big excess of leucines, which wasn't very good. They called it library A. They then made another library where they played with this a little bit. Now they got the leucine amount down to more kind of towards normal proteins. Now, remember, again, I think you probably see where this is all going to go, given what we talked about before. But let's talk about it. So then what they did is they then pieced together, again, this is using old technology, synthetic duplexes that have these QLR sequences, again, randomly dispersed. These are just translating those particular sequences. They also put in a tag, a purification tag, as well as a antibody tag. Which turns out they didn't really use much. Now, the one caveat is that they have some non-QLR parts of this for fitting these guys together. It's really not that big a deal. All right. Excuse me. So, again, they made these proteins. Again, those are the caveats. And when they transformed these into E. coli and expressed them, 5% of them were soluble. Okay. So that got them one paper, like the bugs. And then what they did is they have a second paper where they actually characterized some of the proteins. And so they chose a set of I believe it's 9 proteins from their set. And they purified them and subjected them to structural characterization. A lot like Michael Hecht. One of the first things you should... So what did they do? They did molecular weights. These are the calculated molecular weights. They did guanidinium hydrochloride, denaturations over here. They did melting over here. They did helicity over here using CD. They did oligomeric state by gel filtration. However, one thing you should notice is that this is the concentration of guanidinium chloride that they used in all those experiments. In other words, these proteins really were prone to aggregation like crazy. Okay? So they might have been soluble in E. coli, but when they were purified, they aggregated very badly. Again, nowadays this would get laughed at, but that was the different time. So some of these didn't require guanidinium, okay, but most of them required some, and some of them required 6 molar. One way to look at this, they're ultra stable. They may be ultra stable, but they're also ultra aggregation prone. Okay, so that's that. All right. So there, as I said, there were some that were a little better in terms of their guanidinium, but they didn't really do much to characterize those. So that's kind of odd, and that's because when they melted them, they weren't cooperative. They just got a funny melt, which I'll show you in a second. So the first thing they did, at least reported, was they used melting. They did guanidinium melts and they did temperature melts. And what you can see is that all of these guys have very long melting transitions. They are not real proteins, okay? And in guanidinium, it's the same sort of thing, although some of them look pretty good. One of the problems is that they may not be two-state. And then you have the ones that I was telling you about that were done under zero. They're perfectly soluble, and they have these very weird-looking melts. What this means is there's a whole mess of individual species that are melting, and it just spreads out the melting curve over the entire melting range. So these are definitely not two-state. Okay. But, there's something going on. There's structural changes on the heating. Then they subject them to gel filtration, and pretty much every single one of these guys was a multimer. Okay? In this case, they got a nice clean tetramer, but a monodispersed tetramer, but it was in 3.6 molar urea. So again, it's forming some sort of discrete structure, okay, in terms of its size, but again, it's aggregation prone. And every single one of these guys is like that. NMR, well, there's definitely chemical shift dispersion. So this guy, again, was happy at 3.6 molar guanidine, less than that it aggregates. And this is in the unfolded state with 7 molar guanidine. And basically, you can see the chemical shifts are really piled up here, but they disperse somewhat, okay, when the protein is folded. That's not unique, but at least shows there's folding going on. Interestingly enough, like I said, these proteins were oligomeric, all of them. And when you increased their concentration, you increased their melting temperatures. That is actually exactly what you expect for oligomeric structures because, well, let's do it. I got time to do this. Okay. Okay, this is not going to be a big deal. It's just easier to draw it than to say it. Normally we have this. Okay? And this is delta GF. Oh, yeah, sure. There we go. So let's do this. Okay, the only difference between that and, say, a dimer is you have this. And then there's a dimerization state. So the question is, what's delta GF in this case? Well, unless you have appreciable amounts of this hanging around, delta GF is this. Okay? And that's going to be the delta G of folding of a monomer plus the delta G of dimerization. Okay? So if this is a concentration-dependent step, this one here, it is. It's concentration-dependent. This is monomolecular, so it's not concentration-dependent. This is a concentration-dependent step. As I increase the amount of this around, I'm going to get more of this, and I'm going to have more of a addition of this. So basically, delta GF is going to equal the sum of the monomer delta Gs plus the dimerization delta G. Okay? And that's the term that's concentration-dependent. So that's why these guys are concentration-dependent. And that is a hallmark of multimers. Okay? If you change the concentration and their stability changes, that's because they're multimerizing. It's just mass action. And by the way, this is the same as binding a small molecule. No different. Okay? You bind a small molecule, you will always stabilize a protein. That was one of the questions in your exam. That is true. Because you're always adding the binding energy to the folding energy. Okay. Is that clear with everybody? All right. So that's what's happening here. In all cases, doubling the concentration changes the melting temperature. So they're all oligomeric. And so that's kind of interesting. Why do you think all these guys are oligomeric? I mean, what's the reason? You're taking a random QLR peptide, you're creating them, and most of them form multimers. The ones that have structure. Why is that? What's driving multimerization?
A lot of hydrophobic pressures on the surface, so that's the only way they can wave.
Absolutely. So look, you're taking a random peptide. Okay? You're getting hydrophobicity distributed over this whole thing. 1 in 20 times it makes something that can actually fold. But that doesn't mean it's perfect. And that's also probably why you need like guanidinium to keep in the solution. They've got lots of greasy stuff on their surface. Okay? And so what that tells you is that those are potential sites of multimer formation. And it's actually not so hard to come up with a pattern of hydrophobicity on the surface that gives rise to a defined multimer. Okay? So in fact, that makes complete sense. You would expect that from a random peptide library. And that goes back to our discussions of evolution a long time ago when I told you the first proteins created probably were multimers. Okay? And this is one of the reasons I think that. From a random mixture, it's unlikely you're going to get a perfectly polarized peptide that folds up beautifully, hiding all of the hydrophobicity. Okay? That's one reason. The second reason is kind of what I just talked to you about, is that if basically you oligomerize, okay, in order to fold up, then basically that means the monomers themselves don't have to be as stable by themselves because the oligomerization energy is being added to them. Okay? So to some extent, they both play to the same thing. The fact that you're going to have a chance placement of hydrophobic residues on the surface of an individual monomer promotes its polymerization. Okay? With that in turn, okay, it's going to make the protein more stable. So the fact that the ones that they isolated, which were the more stable ones, presumably, okay, were the ones that could form these defined structures. I think that's pretty cool. So, again, evolution comes along. So you could spend your entire life trying to fish a functional protein out of a library of random amino acids, but we're smarter than that. Okay? And so I'm going to tell you, this is an old paper now, it's 15 years old, which is kind of amazing. But it still has held up to time, and this is a case of, can we design a protein? Now, when I first started doing this class, I said, no way. People have tried, they've tried hard, they've used all sorts of rules, and they were totally unsuccessful. And they were totally unsuccessful. They could make molten globules. That's it. Okay? Jane and David Richardson, who were from Duke University, they've retired since, spent years and years, had a class where they had people design proteins, and every year they'd make one, and it would just be crap. And, it's not their fault. It just turns out it's hard. So we can design a protein, and that's David Baker, who's a famous guy. And if any of you have heard of the suite of programs called Rosetta, it's the only program that can do this, and it's really the best program for doing any sort of sequence-based matching of proteins and structural design stuff. It is the only thing that has survived the test of time. So David Baker started with this many years ago, very bright person. And what he did with his pals is he sat down and sketched a protein on a napkin. That's the story, at least. And the protein had a certain topology. So this is not what he sketched on the napkin. What he sketched on the napkin is here's a strand, here's a helix, here's another strand tied to it, here's a helix that goes underneath, and then we'll have another turn and another strand, another turn and a helix, and a turn and a helix, and so forth. And we'll have these guys pairing together to form a beta sheet. So basically it's a beta sheet platform with two helices attached. A fold that does not exist in nature, at least in theory. Okay? And then they fed it to Rosetta. So what does Rosetta do? You really need to read this paper if you want to understand Rosetta. I could give a whole class in trying to explain what Rosetta does, but I'll just try and break it down into the bits. Okay, and the bits are this. What Rosetta does is it goes through the protein data bank, or has already gone through a protein data bank, and collects pentapeptides, all the pentapeptides that are in the protein data bank, so including the overlapping ones, and characterizes them conformationally. So the first thing that happens is you just take this napkin design and you go to a computer and build a model that's protein-like. Has helical geometry, has all the right Fifes angles, and so forth. And you kind of slap it together. So you build a preliminary model. Computer could probably do that, but a human can do it pretty quickly. And then you feed it to Rosetta. And what Rosetta does is it goes through that model and says, okay, through my whole bank of 100,000 pentapeptides, which one of them fits this one best? Okay? And it takes the sequence from that and sticks it into that position. Okay? And it does that about 100,000 times. And then it goes through that and says, well, which one of these makes any sort of energetic sense? And it turns out that the easiest way to determine whether it makes energetic sense is just simply to energy minimize it. And that's what they did. And so now you take your 100,000 and you pare it down to like 100. Or maybe it's 10,000. Okay, you pare it down to 100. So you take the top 1%. And from there you have sequences. And so that basically erases everything that's got crazy stuff like very polar groups and so forth. Well, if you try and make something there, you end up with, so you have 100 sequences. They're all garbage. Okay, they don't do anything. So then what Rosetta does, and this is really the brilliance, is it then starts to mutate residues, okay, and basically change things around one at a time, and also goes through another rebuilds. The first step before it does the mutation part is it goes through and rebuilds, again, because energy minimization has changed the shape of this thing. It goes back to the peptide library and rebuilds it from peptides all over again, expanding that 100 sequences back to 10,000 or 100,000 sequences. And in the process also generates mutants at different sites, iteratively, going through entire sites. It's very computationally expensive. It still takes like weeks to do this. And then it recalculates energies of that 100,000, cuts them back down to 100. And it just cycles through that, essentially, until the energies of the structures that it gets, the calculated energies of the structures that it gets, don't improve. So then they stopped, and this is it's not a swindle, okay, but it's just the part that people don't realize. They made 100 of the top scoring proteins. 3 of them folded into real proteins. Now you may say, 3%. I say, holy jeez, that's amazing, okay? You have a structure that you've basically drawn out on a piece of paper, okay, and now you have 3 out of 100 proteins that actually work. That's phenomenal. That's phenomenal. And not only do they work, they really work. When I first read this paper, I said, this has got to be a lie, okay? But I know David Baker. I said, he didn't lie, okay? But I said, this is just too good to be true. This is showing the fit between their calculated structure and their X-ray structure, which they were able to get. First time anybody had ever gotten X-ray structure of a designed protein. The melting temperature of these guys are like 95 degrees, okay? They're incredibly stable. And I don't remember which is the calculated structure and which isn't because it doesn't really matter. They're the same, okay? So, bam, David Baker solves the problem. Phenomenal, yes?
When you're saying that the protein works, like what?
When I say it works, it has all protein-like characteristics. Okay, it has thermal melts that are completely cooperative. Okay, very high stability, very high delta G of folding. So, and two-state behavior. Yeah, so that's fine. Sorry. Okay. Here's the problem. This is true for all of these sorts of things. Is that we don't know why. We can't extract the kernel, okay, from this and say, here's the thing that we've been missing all along, okay, that tells us now how to do this. If you ask David, what he says is, well, what we're doing is filtering out all the garbage by using the protein data bank. Okay? We're taking things that are known to exist already in known geometries, okay, and using that as our basis set. That would be his explanation. But in terms of what the meaning of that is and how you could make a de novo design program that didn't use protein data bank information, it's not possible. Okay, yet.
When was that paper?
2004.
So, I mean, they're using machine learning, right? That's the whole.
The machine learning part came later. But no, originally there was no machine learning. It was all iterative.
Was it like an early form of machine learning, or just?
No. It was brute force. Brute force, bang and select. So it's an evolutionary algorithm, right? You're generating variants. Okay, you're selecting variants that are at the top of the heap, and then you're using that as a seed for your next set of variants. And so you just keep going down through there. So, I mean, the thing is it doesn't require that energy minimization is precise because you're really not using it that way. You're not using it to evaluate one structure. You're using it to eliminate bad structures. So that's really the key. So, fantastic, Brave New World. David has gone on to use this program to do lots and lots of things. Oddly enough, I haven't seen too many papers where people have done de novo design since then. But people have certainly used it to operate on regular proteins. And, all I can say is that it's certainly better than the tools that were there before. And it is really the only successful case of de novo design. It's interesting because there was another guy whose name I won't mention, he was evil, who was lying about doing this, okay? He had kind of a similar protocol, but it was always kind of half-baked. And I'd seen him at conferences a couple of times, and I said, this guy has got to be full of it. And it turns out he was completely full of it. So he lost his job. Okay, so really the kind of take-home from this lecture is, first of all, the hydrophobicity patterns are the most important aspect of a protein that determine its fold. Okay, that again, you constrain the number of conformations available to you hugely, okay, by just having a very particular hydrophobic residues. And if you can't find a structure that does that, okay, then you don't fold. Simple. Okay, so the structures we see are ones that have been able to use the hydrophobicity pattern to fold. And then one of the questions is, well, how many folds are there in the universe? And the answer is probably a limited number. There's been a number of computational experiments to address this. Certainly the protein data bank, there's still quite a few proteins. If you look at, say, sequence patterns in all of the known sequences now, we still don't know the structures of most of the things out there. But it's true that the number of unique folds that are solved every year by crystal structures has been going down over time. Okay. So how probable is it for a non-designed protein to form something folded? Not so rare. Not crazy. Okay. I mean, 1 in 20 is probably a little bit exaggerated. But, even if it was 1 in 1,000 or 1 in 10,000, that's pretty good. From an evolutionary standpoint, you're usually working with billions and trillions of possibilities. And so, that kind of probability looks pretty good to me. And do we know enough to design a fold? We don't know enough, but we certainly can use existing structures in a clever way, okay, to design proteins that have new folds. Okay, so that is the end of protein structure. Any questions? Yeah?
So you said was it calculated the most stable? I know that you said it's calculated as evolutionary checklist, it's the most stable is what we want, but I know that you said previously that the most stable is not so the most.
I'm sorry, I'm having trouble hearing.
Sorry, you said, so last time you said that the most stable is this Rosetta selected the most stable form of the protein. But
No, what I meant was excluded. What I said about Rosetta is what it does is it uses the energy function to exclude all the bad ones. Does that make sense? So rather than when you're doing a design, okay, and you're generating lots of variants, you're trying to essentially, what you're really doing is exploring the possibilities, right? So you generate this huge set of variants either by rebuilding, okay, or by mutating and then going through an energy minimization, reminimization protocol. So it's a cyclic set of versions where you're generating variants and evaluating them and deciding whether to keep them or not. So in the context of that is, what's your question?
I guess, so I'm trying to wrap my head around what you said about protein folding being the most stable, or at least what...
Oh, sorry. Yes, the form that we form is the most stable one. That's right.
Yeah, so in earlier lectures you said that the most stable, not to say the best, for that.
That's right. Well, no. In fact, I think that the Rosetta proteins are over-designed. I mean, they have melting temperatures that are way high. Okay, so in fact, I think that kind of shows you that probably the wild-type proteins aren't the most stable possible fold. The evolution so let's take a step back. The evolution that's going on in reality, it's got to be good enough, right? So I think what we've learned is that good enough is up to 20, 25 kcals per mole. Okay, but Rosetta doesn't have that limitation. It's just going for the lowest. And so it generated something that's really, really stable. Okay, does that make sense? Yeah. And what's interesting, I have to say, pretty remarkable is that they did get salt bridges out of the design, which is, again, not been able to be done by, say, mutational design, but just simply sticking two pairs of oppositely charged resonors next to each other. That doesn't work. It requires more than that. Other questions? Okay, that was a lot to digest. I think I'm going to, actually went rather quickly. I was kind of surprised. But do expect to see the stuff in the exam. Please do the rest of the homeworks that are relevant to this. So the rest of problem set 3 from 6 to 9, I think is relevant. Everything having to do with protein folding in vitro and in vivo is not. I could sort of start talking about that, but I feel like that's a lot of stuff we've done already. So I'm going to hold off on starting the next... Well, should I do that? No, I'm not going to hold off. And the reason is that I forgot that we're also going to have to go over our exam when we get it back. And so I want to make sure we have time to do all of that. Okay, wait, don't move. We have 20 more minutes. I know, I shouldn't have said anything. Okay. Let's completely switch gears because I like talking about protein-DNA interactions. So, we've talked about protein folding. We've talked about nucleic acid structure. We really didn't talk much about nucleic acid folding. I'm sorry. We just haven't, the course has never had time to cover all of that. But I'll just simply say that in the case of nucleic acids, the driving force is not the hydrophobic effect. It's what? Hope you guys don't forget that. So hydrophobicity is definitely driving the fold of a protein. But what's driving the structure of nucleic acids to fold? You guys have forgotten this already? Stacking, thank you. Space stacking and hydrogen bonding. Okay? Hydrogen bonding is driving the specificity of structure, and base stacking is also doing that. So again, it's kind of the same sort of deal. In the case of nucleic acids, though, the hydrogen bonding pattern, okay, is obviously driving the specificity, much like the hydrophobic effect is driving the specificity of proteins. So even though energetically hydrogen bonds aren't really giving you much, they're basically allowing the formation of certain structures over all others. They're allowing the differentiation of those. So there's some fundamental differences. There's also no big delta CP for nucleic acid folding, but there is for proteins. So that's because there's no hydrophobic effect. Okay. So now we're going to kind of merge these two things together and talk about how proteins recognize. And we're only going to talk about DNA. Everything I'm telling you would be true for RNA too, but RNA is a little bit more complicated and there's a lot more variation. But a lot of the same things I'm going to tell you about here are true for RNA-protein interactions as well. So one of the things that we have to think about, because I'm a bioenergetics type of guy, is that there's these terms that are thrown around that we really need to talk more about. We start talking about protein and nucleic acid interactions. And one of them is the idea of affinity versus specificity. The second thing is that there are proteins that are specific in binding DNA and the ones that are non-specific. And so how do we think about that? How do we measure binding? We haven't really talked much about methods, but here we're going to kind of get into the nitty-gritty of it. We're going to find out how proteins recognize DNA, and there's sort of three different fundamental ways they do that. They recognize the backbone, they recognize the bases, and they also recognize the ability of the DNA to change its structure. If you want to make a DNA binding protein better, what can be done? And nature has done a bunch of things that we can learn about. How does a DNA binding protein find its site? Okay, and this 1D diffusion idea turns out to be wrong, okay, but we'll talk about it. And then finally, if I have a protein and I want to know what DNA binds to, how do I do that? What's the methods I would use to find out about that? And I did just forget that I did not give you a break. So I'm not going to go very deeply into this. I'm going to go one slide in, then we'll break. So I apologize for that. I saw people walk out and it's like, oh yeah, I guess I didn't give you a break. You're probably tired. All right, let's talk about affinity and specificity, and that's a good place to leave it. So affinity, folks, is just like stability. It's a delta G. Okay? But typically we don't measure it in kcals per mole. That's just a matter of convention, because K and delta G, as we know, are related directly, by log. Okay? So when we talk about affinity, we usually do it in the form of a dissociation constant. And so if we have a protein and DNA pair and we have a protein and DNA complex, we're really actually looking at this direction. Okay? It's just like the unfolding thing all over again. In this case, I like using KD, even though it's the opposite direction one would go in, because the units of KD are useful. They are concentration units. Okay? Now, Ks are, in theory, unitless, so this gets into a complicated philosophical discussion that I don't want to go anywhere with, but just let me tell you that the KD, whether unitless or not, it is in concentration units. It can be thought of in that, and that's a useful thing. So just, hopefully you had this when you were in 102, but what is the use of a KD? If I asked you for a functional definition of KD besides the equilibrium constant part of it, what would it be? Yeah.
Rather, if the molecules prefer to be together, reactive,
Okay, but I'm talking about in a quantitative way. Quantitatively, if I gave you a number, what would be the meaning? If I told you the KD was 100 nanomolar, what would be the use of knowing that? Under what conditions, though? Yeah, that's not quite useful. Yeah.
The substrate concentration, which is half saturated, half unsaturated.
Correct. That's the use of a KD. It's the concentration of free ligand, okay, at which you're half bound. That's useful because it's telling me at what concentration range, at least what order of magnitude I have to be concerned about change. It's pretty much an order of magnitude below the KD, you're not bound. An order of magnitude above, you're fully bound. And everywhere in between is where you're concerned about. That's useful, okay? Because it tells you, and to get 10x KD, that's when you're at 93% saturation, okay? So it gives you a concentration ranges over which the action is occurring. And that, obviously is useful. If I'm designing drugs and I come up with a micromolar drug, it's never going to be useful. Okay, because I couldn't get high enough concentration inside of a tissue or in blood without killing somebody for the most part. So pharma, when they're designing drugs, they want to see nanomolar KDs at the outset. If they're not starting with something nanomolar KD, it doesn't even bother. Okay, so KD has a very useful number, and again, it's the number which you're half saturated. That is a very useful thing to remember. It's the analogous of Km, okay, from enzymology. All right, it's analogous to that. So if you're cool with 123, then this should be no problem for you. And again, since it was done in 102, I didn't want to kind of repeat it. So KDs are useful, and that's how we characterize affinity. Specificity is different. And the reason I bring this up is that you see, especially in the old literature, where people say, well, if it's high affinity DNA binding protein, it must be really specific. And that's a completely fallacious statement. Okay? There's no relationship between specificity and affinity. What specificity is, from a quantitative view, is the difference in KD comparing two different ligands. That's specificity, or two different proteins, however you want to do it. So it's a ratio. In which case, if you're looking at how a protein distributes itself between two different DNA sequences, that's what we're interested in when we talk about specificity. All right? So automatically, when you say specificity, if you really mean it in a quantitative way, you have to have a reference for what you mean by not specific. And that's a big deal. And that's where I'm going to leave it, okay, because now we're at 10 minutes, and that's my usual break time. We'll come back and take this up on Monday. Have a good weekend. Hopefully we'll have your exams.